Hi, my name is Clayton Brady. I am going to be a sophomore here at Brockport. I'm a biology major, and I have been working in Dr. Rich's lab for about 10 months or so. I started as a freshman and carried through first and second semester, and now I've been working here all summer. So the overall goal of this lab is to better understand the regulation of GI motility. Specifically, we are studying the gene anoctamin 1 and anoctamin 2. We know that these genes are expressed on these specialized cells called ICC cells, or interstitial cells of Gahal. We know that these cells are involved in GI motility as pacemaker cells, so they sort of develop the spontaneous rhythmic contractions and play a large role in those contractions, which help with GI motility and help the regulation of GI motility. So we're trying to better understand this regulation. We know that anoctamin 1 is a calcium-activated chloride channel, and we know where it is. We're just trying to understand how it is involved with GI motility. So my part in this research experiment, as a new, fresh researcher, haven't been around too much, is to generally just keep track of the fish and take care of them and feed them and just help with the general upkeep in the lab. And also, I've played a big role in the Morpholino oligonucleotide injections. And Morpholino oligonucleotide, or MO, is what we use to knock down the specific gene we're looking at, or the gene of interest. And we do that by injecting the embryos with MO. Normally, the embryos are only about 20 minutes old, and it is important to inject them around then because they haven't reached the 2 to 4 cell stage, and once they reach that stage, the MO isn't drawn into the cells as well. The first step in doing microinjections is to pull your micropipette. It's basically a fine glass needle that gets pulled apart to a very, very fine point, which then we clip, and we use that to inject the embryos. So what we have is a glass needle, or actually this is a glass tube. It's not quite a needle yet. We put it into the puller, and the trick is to push it in between this filament. And this filament gets very, very, very hot. And as it gets hot, it heats up the glass, and compressed air pulls the glass tube apart, which pulls the middle of it into a very, very fine point. The first step of the process of this experiment is injecting the embryos with Morpholino oligonucleotide. MO is a chemical that we use to knock down gene expression, and we're specifically knocking down the gene of interest, which is anoctamin 1. So we pulled a microelectrode. It's a very, very small glass tube, which I will show later how we actually pull it. And it pulls the tube into a very, very fine tip. And this tip is what we use to stick into the yolk sac of the embryos and inject about 0.036 picoliters of the Morpholino. Once it is in the yolk sac, it is then drawn into the cells as they begin to divide. This is how we introduce the chemical into the cells, and as the cells replicate and the animal gets bigger, it knocks down the specific gene in each of those consecutive cells. So the process, like I said, is to pull the needle, and we then harvest the embryos from the fish room, and we hustle them back to the lab because you have to inject them around the 2 to 4 cell stage, because once they get too old, it won't get into all the cells. So that's about the stage that we're at now. We line them up in a plate mold that we made a few days before, and we do the injections. So we have the manipulator, which holds the microelectrode, the small glass tube, and we have a tube that is attached to an air compressor. And what happens is that we force air through the tube, and it pushes a specific amount of chemical into each embryo. So the trick to doing this is we have a foot pedal that we use to supply power to the air compressor. And we look through the microscope, and we line up the needle with each embryo. So the trick to injecting these embryos is to line the needle up with the embryos, and then we push it through the chorion and through the yolk sac. It's important to make sure that it goes all the way into the yolk sac and not just through the chorion, because the MO will not be drawn into the consecutive cells as they divide.